Hematology, oncology, physiology. So the first thing we need to talk about for Heme Onc physiology is the antiglobulin test, which is also known as the Coombs test. And all that the Coombs test does is just detects whether there's antibodies against red blood cells within our blood. Now, there's two forms of this test. There's the direct and indirect form. Direct Coombs test just tests if there are red blood cells that are coated with antibodies. So if the antibody is directly attached to a red blood cell, then that means that the direct Coombs test will be positive. And we use this direct Coombs test to diagnose AIHA, which is autoimmune hemolytic anemia, which we'll discuss later on in the pathology videos. Secondly, the indirect Coombs test, which is not as important, just is used to detect whether there's antibodies within the serum, but not directly attached to the red blood cell. Moving forward, I'll be talking about primary hematostasis, which is just a platelet plug formation. All this is interaction between platelets in the vessel wall, as well as platelets with each other. The coagulation cascade has nothing to do with primary hematostasis, and that is known as secondary hematostasis. So, to begin primary hematostasis, the first thing we have to understand is that endothelial damage occurs. So once we have endothelial damage occurring, it's just the vessel wall breaking, something occurs like that. The first thing that occurs is vasoconstriction, because we don't want more blood going to an area that is more damaged, causing more leakage of blood. So vasoconstriction occurs first. Next, we have von Willebrand factor, which is present in the serum. It comes and it binds the exposed collagen, which is collagen type 4 at the basement membrane. Once von Willebrand factor binds, platelets that are present in the serum come and bind to the von Willebrand factor via their GP1B receptor. So platelets bind to von Willebrand factor via their GP1B receptor. Next, after this occurs, the platelets release ADP and TXA2. TXA2 is important for vasoconstriction and platelet aggregation. However, ADP is more important because it binds to the P2Y12 receptor, which induces GP2B3A receptor expression at the platelet surface. And GP2B3A expression allows for platelets to bind to one another, leading to platelet aggregation. Once this occurs, fibrinogen also binds GP2B3A receptors to link these platelets. And fibrinogen comes from an inactive form known as fibrin. With all this together, this leads to the platelet plug formation, and that finishes our primary hematostasis. Now, just as an overview, what occurs first is endothelial damage, then vasoconstriction, then von Willebrand factor. Platelets come bind von Willebrand factor via GP1B, which releases ADP, allowing for GP2B3A to be expressed, which makes fibrinogen in order to bind the platelets together. So I want to be clear that GP1B and GP2B3A are both located on the platelets. However, GP1B binds von Willebrand factor, whereas GP2B3A binds fibrinogen, which allows for platelet to platelet aggregation. Next, the coagulation cascade occurs due to the fact that primary hematostasis is weak and unstable. So secondary hematostasis must come in and allow for a better clot to form. The coagulation cascade is broken up into two pathways. The first pathway is the extrinsic pathway, and the second pathway is the intrinsic pathway, which come together at the end to form the common pathway. The intrinsic pathway begins with factor 12, and then progresses to activation of factor 11, and then factor 9, then 8, and then 10, which is a part of the common pathway. The way that people remember this is just by using the mnemonic TENET, T-E-N-E-T, 12, 11, 9, 8, 10. Now, one specific thing that we have to know about the intrinsic pathway is that factor 8 is carried by and protected by von Willebrand factor. So not only does von Willebrand factor affect platelet aggregation, which we talked about in primary hematostasis, but it also affects the intrinsic pathway due to the fact that it carries factor 8. That's a question that they love to ask about, so make sure you understand that if patients have a deficiency in von Willebrand factor, which we'll discuss later on in the pathology videos, you have to understand that PT, which is what we use to monitor the extrinsic pathway, and PTT, which we use to monitor intrinsic pathway, can be changed due to the fact that von Willebrand factor is decreased. Now, once again, it's PTT that will be affected if we have a deficiency in von Willebrand factor, because factor 8 is only located in the intrinsic pathway. Okay, and we'll get to there soon, but I just wanted to kind of give a heads up because that's a question that they love to ask. Moving on to the extrinsic pathway is just factor 3 and factor 7. Honestly, they never asked about factor 3, so understand that factor 7 is the first thing, and it goes right into factor 10, which is a part of the common pathway. So just understand that factor 7 is really the only factor that you should really care about for the intrinsic pathway. Now, once we finally reach that factor 10, it has to be used to activate factor 5. Okay, so factor 10 is the common pathway, and then factor 5 activates it. And once we have that factor 5 and factor 10 activated, it allows us to activate factor 2, which is known as prothrombin. Prothrombin activated turns into thrombin, and that is a very important aspect of our clotting because thrombin is what actually clots for our coagulation cascade. Because thrombin activates fibrinogen into fibrin, which leads to the aggregation of platelets, which is the most important part, leading to the fibrin mesh stabilization. So for an overview of the coagulation cascade, we have the intrinsic pathway, which is monitored by PTT, and it goes factor 12, factor 11, factor 9, factor 8, and then factor 10. Whereas the extrinsic, which is monitored by PT, INR, contains factor 7 and then goes to factor 10. And then the common pathway, starting with factor 10, then factor 5, activating prothrombin into thrombin, and then thrombin activates fibrinogen into fibrin. The last thing that we need to understand about the coagulation cascade is that we have two regulatory molecules known as proteins C and protein S. Both of these are going to be inhibitors of coagulation, leading to an anticoagulation state. Another protein that works against the coagulation cascade to favor anticoagulation is plasminogen, and plasminogen is the inactive form of plasmin. And the way that we activate plasminogen into plasmin is we use TPA. TPA is just a thrombolytic, and it is produced in the body, but we can also provide it extrinsically as a medication. All that plasmin does, once activated, is that it breaks down the fibrin mesh that is stabilizing the platelet plug, leading to fibrin degradation products known as D-dimer. D-dimer is a very important marker in the pathology of DIC, which we'll talk about later on. Lastly, in order to make this all fit together, we need to talk about vitamin K's role in coagulation. So vitamin K is used in order to synthesize factors 2, 7, 9, 10, protein C, and protein S. And the way that it does this is that it uses epoxide reductase, an enzyme, in order to reduce vitamin K, which is its active form. Once vitamin K is activated, it can be used for gamma carboxylation in order to activate our factors 2, 7, 9, 10, protein C, and S. And one medication that blocks this is going to be warfarin. Warfarin inhibits epoxide reductase, leading to an inability for vitamin K to become in an active form, leading to an inability for gamma carboxylation to occur for those specific factors and proteins. Other times we will see these factor deficiencies for 2, 7, 9, 10 is in neonates who lack the GI bacteria which produce vitamin K, as well as patients that take broad-spec antibiotics, because broad-spec antibiotics kill that bacteria that once again produce that vitamin K, which is needed for coagulation factors 2, 7, 9, 10. Another question that they would love to ask is that factor 7 has the shortest half-life and factor 2 has the longest half-life. Lastly, this whole entire concept of vitamin K-dependent coagulation takes place at the liver because epoxide reductase is only located at the liver where vitamin K is activated.